of the IgG class, number of binding sites. All of that refers to is how many hands are available on each antibody molecule that could bind antibody. So there's only 2 hands on an IgG because it's a monomer, and so we have 2 antigen binding sites. And the molecular weight, you can see that when you compare it to all of the other antibodies, it's the what? It's the lightest, or the smallest. I want you to think about it in your mind, stick it in there. Not only that it's molecularly the lightest, I want you to think of it as the smallest. Because the smallest one would have the easiest time crossing a placenta. So when you think about, which one of the antibodies crosses the placenta? Well, it's probably the smallest one. The placenta is we have this vascular system, and it's got to be really tiny to get across. So you will remember 2 things at one time. The molecular weight is the smallest. So it's the lightest, it's the smallest, and because of that, it's best suited to slip across the placenta from mom to baby. We already said that if we drew our blood, we'd see that most of the antibody that we have in our circulation is IgG. And that's another good thing for the baby, because we know that things move from areas of what? Higher concentration to areas of lower concentration. So if we got a huge amount of something on one side, it's going to be easiest to move that stuff to where it's just a little bit or nothing. So another good thing for the baby is that mom's got so much of it in her circulation that the baby will stand a pretty good chance of getting some of it to move across to its circulation, because it's not doing a very good job at making antibody because it's not being exposed to antigens in the perfect world. Another good thing about IgG is its half-life. Its half-life as compared to everybody else is what? Longest. Wake up. I'm going to start serving coffee. So it hangs around the longest. So that's good for the baby, because when the baby comes out, it's going to be working only on what mom has given it. So good thing it's going to hang around a long time, because after 23 days, it's going to still have half of the mom's IgG left. And so after 23 more days, it's going to cut in half and half and half. So after, a few months, the baby's not going to have any of mom's left, but it's already going to have a few months of making its own. So that's a good thing for the baby. So IgG has there's more of it. It's tiniest, and it hangs around the longest. Crosses the placenta, yes. Fixes complement, yes. IgG, and what fixes complement? What's the other one? Quick. Huh? IgM. Right. Binds to phagocytes, yes. And the functionality, it gives us long-term immunity. It hangs around a long time, so that if we had chickenpox when we're 5, when we're 50, we still got some around, and it's but we have the memory cells that's ready to squeeze out more, so we're never going to get it again. It neutralizes toxins and viruses, and we'll talk about how that happens as we move along. Okay, we know that IgA is a dimer, but it can where is it a monomer?
In the blood.
In the blood, in the circulation, it's a monomer, but even when it's a monomer, it's bigger than IgA. It's next highest in concentration in the circulation. All right, here's a don't let me trick you on this. How did you guys do on the stuff I told you not to let me trick you on? Who missed something I told you don't let me trick you? me and my study partner, we made a whole sheet. We had a whole page of "Don't let me trick you."
It was Esther and I, and we like made this whole thing of like, do not let me trick you, and I'm still different.
She goes, I can't believe I knew it, but oh well. All right, don't let me trick you on this. IgA is second highest in concentration in the blood, in the circulation, and the serum. But fascinating, important, easy to forget feature of IgA is that in the entire body, if we look at the entire body as a whole, there is more IgA in our body than any other isotype. Because we have so much serous cells and so much mucous membrane in our body, the GI tract is cranking out secretory IgA all the time. So, if I ask you which isotype of antibody is present in highest concentration in the circulation, your answer is IgG. But if I tweak the question and say which isotype of antibody is present in the highest concentration in the body, you will say IgG. Don't let me trick you. All right, very good. Feel sorry for people who have not in their chairs right about now. But okay.
No mercy.
All right. Half-life is pretty short, but that's okay because the mucous membranes are just cranking it out. They're just turning it out every day. They're making more and more of it. So it doesn't have to last a long time because so many cells are making it constantly. Crosses the placenta? No. Fixes complement? No. Binds to epithelial cells, and that's a good thing because the cells that line our gut that's making all of this stuff, it's a good thing that it has these antibodies in there. So we always having some protection against stuff that's coming through. We got some hands sticking out saying, nah, you're no good, and we're gonna sort of take you out and get you, drown you in all this serous mucousy fluid. Okay. IgM, huge. Look at that molecular weight. That's crazy. How many antigen binding sites? Well, there's 10 of them because it's a pentamer. It's 5 of them. Each of them's got 2 hands. So we have 10 antigen binding sites. Average time in the serum is pretty short. But that's okay too, because we said that IgM is the first class of antibody that's made when we are first exposed to an antigen for the first time. It's okay that it doesn't last long because as the concentration of IgM is going down because of its half-life, the concentration of IgG is going up because we're going to start making IgG and IgM becomes less important to the defense or the protection of the host. Crosses the placenta, no, way too big. Fixes complement, yes. Doesn't bind to any of those cells. What's its claim to fame? First responder. And we also know that it can be B-cell receptor as a monomer. IgD, we know that its main job is to be a surface on the surface of B cells. And IgE, we know it's the allergy and the helminthic job that it performs. We also know that it stands on top of mast cells and basophils and eosinophils, and it is important in that hypersensitivity or the degranulation of the granulocytes in the inflammatory process, and all those allergic reactions as well. You see, there's very, 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 very little of it in the circulation. It's going to be higher in people during, pollen seasons and all those things that may cause their allergic symptoms. There are more of those types of antibodies around. Okay. In serum, we know that the immunoglobulin is made primarily of IgG, and most of the gamma globulins, we call them gamma globulins because antibodies pretty much electrophorese out in the gamma band. So that's how they got sort of referred to as gamma globulins. But they separate out into alpha, alpha 2s, betas, and gammas. But most of them are separate out into the IgG because there's more IgG in the circulation. Okay, these are all the things that we know already. Feel really smart when we're going through these. You know all of this stuff already, nothing new. IgG. Again, we know that they can attach by their feet to phagocytes. We talked about this natural killer cell thing. We'll talk about that still to come. Antibody-dependent cytotoxicity is what natural killer cells do, and that is an antibody-mediated killing or antibody-mediated cytotoxicity, and it's mediated by IgG. The FC portion is the portion of the IgG, the little bitty light IgG that enables it to cross the placenta. It goes in feet first, across the placenta and into the fetal circulation. Feedback inhibition of B lymphocyte activation. What in the world is that talking about? Some of those fancy words again. High levels of IgG feedback to B lymphocytes prevents their activation and return and blah, 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 blah. So what does that mean? In plain old ordinary language, when the body has responded to an invader and it's turned on the system that makes antibody, once the antibody titer gets to a certain point, the antibody itself acts as a feedback to say, B cells, stop, hold everything, we got enough, you're wasting time, you're wasting energy, we don't need you to keep doing that. So, the antibodies that are made by the B lymphocytes of the IgG class, when the titers get to a certain point, the titer itself, the concentration of IgG gets to a high enough level that it turns off its own production. Okay, so it feeds back to the B cells to say enough. Okay. There's our IgM, our big pentamer. When it's on B cells, it's a monomer. They're hooked together by their feet by a J chain. Disulfide bonds between them. Now we haven't talked about this. The property of IgM that makes it really special is its ability to agglutinate microbes. Now, it makes sense that they would have this special characteristics because they got all these hands. Okay, so we're exposed to some microbes, and we said IgM is the first type of antibody that's produced. Doesn't it make sense that this type of antibody would be really good at agglutinating the offenders, the intruders, because they got so many hands, they can hold on to the microbes. Another M pentamer is here, and it's holding on to stuff, and everybody's holding on, and everybody's all the microbes are agglutinated. They're held together. They're locked down by all of these hands grabbing them, and they kind of keep them cloistered so that they can't get away, they can't get out, they can't move into other parts of the host to make the host sick. Okay? So that's one of the great features of IgM. It's very good at agglutinating because of the way that because of its structure, because of its anatomy. And when it holds the microbes, the rest of it knows the phagocytes are coming. So they just hold everybody together by their hands. Everybody's grabbing something that's specific to that molecule, and it holds them while the phagocytes come, and the phagocytes can come, and it makes it really easy. So it kind of is has an opsonizing effect for the phagocytes who are coming. The first class of immunoglobulins produced in the fetus, when the fetus is making its own antibodies, the first kind it's going to make is IgM2, because they have the same type of immune function as we do. Mostly the fetus is not going to be producing anything because they're not being exposed to anything. The unfortunate fetus who's had a virus or something that crossed the placenta to infect the fetus will, depending on the age of the fetus, the maturity of the fetus, the maturity of its immune system, if it's capable of producing antibodies, IgM is the first it's going to produce as well. And we said the half-life is pretty short, but usually that's okay. 10 epitope binding sites, 5 of them, 2 hands on each. Able to activate complement. We said that both IgG and IgM can activate complement, the others don't. But in red letters it says, and don't let me trick you, because we have this idea that IgG is so important and there's so much of it around. But if we're talking about efficiency and activating complement, of course it would make sense that IgM would be the most efficient complement activating, classical complement activating molecule, because it's got so many sites to do it with. And so it's really, really good at activating the classical complement pathway because it comes in 5 pieces. It takes five molecules of IgG to do the same work. So we know that they both activate complement, but if I want to know which one's better at it, you're going to say
IgM.
yes. Okay. And we know all about the last one already. We said that a quadrillion times. Okay, there's our dimer, IgA. It's going to be in the dimeric form in mucin and serous and tears and all the body fluids, saliva. And we have that secretory component that's associated with it to make it more stable in those secretions. And the half-life is very short. We know that in the circulation, there's not a whole lot of it around, but in the body, there's just a huge amount of it around because there's constant turnover in all the mucus and serous cells in our body. So bunches of it when we're talking about the body. There we go. All that red stuff means you let her trick you. 6% in the serum, produced in the largest quantities when we're talking about the entire body. In fact, day, humans secrete 5 to 15 grams, that's a whole lot of grams, a whole lot of antibodies into their mucous membranes every day. Okay? Mostly associated in mucosal cells, malt tissues. You know it's a dimer, you know it's got 4 hands for binding antigen. Has a secretory component to keep it safe in all of those fluids where they hang out, bless you. The third one is kind of interesting. The secretory IgA binds to components of the mucous membrane and contributes to the ability of the mucus to trap microbes. So mucus is pretty sticky and gooey, and that's a good thing. You know, that's one of our innate defense mechanisms, just having goo and sticky stuff to hold on to things that are trying to get in. But having the antibody present in the goo makes the mucus even more efficient in trapping microbes and not letting them go. So the IgA is going to try to situate itself along the Fc portion of the mucous membrane so that its hands are available to trap microbes and keep it locked in the mucus. Can activate the alternative complement pathway. Now I put that up there and I wrote the little green notes because if you start looking around on the internet, and I imagine you do, stuff gets, piques your interest and you go, well, I wonder how that works. And you Google something and you find that, oh my goodness, she said that only IgG and IgM activate the classical pathway. And I say, yeah, that's right. But you'll say, you'll read something and it say that IgA can activate complement. Make sure you read this part. It's controversial. You'll see it in multiple places. But even if it can, it's not activating the complement pathway via the classical pathway that we've learned about. It's some sort of alternate way that it's getting the complement cascade to go. So still, even if you've read that, still, if I ask you which will activate the classical pathway, it's still going to be right when you tell me what? IgG and IgM, right. G, monomer, tiny amount in the circulation. Where it is, it's mostly stuck on cells, and important on B cells to get the whole activation of all the things it does, endocytosis, presenting and activating, presenting antigen for the helper cells, and then getting everything just to cascade into to all the things that happens once the cytokines are released. Half-life is short. Monomer to epitopes. Okay. That half-life is the half-life that we're talking about in the circulating concentration that we're talking about. We're talking about it in the circulation. Where it's important for it to be more stable is when it's actually on the B cells and acting as receptors. Surface of B cells, where they control B lymphocyte activation and suppression. Now, we said that high levels of IgG can act to feed back and inhibit more activation. So that's what this is saying, that the presence of IgD can be a part of that link, a part of that system, that feedback system. They're sitting there, but when lots of IgG comes back and starts to talk to the B cells and say, okay, that's enough, IgD on the surface plays into that in some way. may play a role in eliminating B lymphocytes, generating self-reacting autoantibodies. Well, that's something that we may not know the full mechanism for how it happens, but it's thought that when antibodies are formed that have the ability to recognize self, IgD may interact with those lymphocytes that are doing that to say, okay, no, this is not good for our host. So it may have some immunoregulatory effects as well. That's just serving as a receptor. Really little bits of IgG in the circulation, unless we're actively responding to something, like we have a parasitic worm infestation, then we expect that there's more IgG in our circulation. Half-life is very short. These are the antibodies that are involved in allergic reactions because they stand on the granulocytes. And what happens is, let's say I'm a mast cell or an eosinophil, and I have IgE standing on my surface by its feet, and I'm allergic to something, okay? And I'm allergic to this thing that my antibody will recognize if it comes. If I'm feeling great right now, but pollen season is coming, and my eosinophils are just kind of hanging out. They're not doing much, but they have these antibodies on their surface. Now I breathe in these antigens, and my hands grab a hold to these things that I'm allergic to. Once they bind, the message goes to me, I'm a mast cell, that these things are in the environment. In fact, you have now attached, your antibody has attached to it. Degranulate, start putting those cytokines, all of those histamines and all those things that you got in your granules, dump them out, okay? So that's how the IgE is active or interactive or required for these allergic reactions. They stand up on the granulocytes and just kind of wait for the thing you're allergic to come near. It grabs it, and it causes the release of all the allergic mediators. Okay, we'll talk about some more of that stuff later. FC, that's just what I acted out just now. Cross-linking of cell-bound IgM by antigen triggers the release of vasodilators. So what that's talking about is that if I'm an IgE and this is an IgE, and we're both standing on top of a mast cell, it notices that our antigen has come. I notice that the antigen has come. This is the cross-linking that is somewhere. It's talking about cross-linking. Where is it? It's that cross-linking of IgE bound. And so once we have cross-linked, we both together give the signal to the cell we're standing on to release vasodilators and all those histamines and products of inflammation. FC portion of IgE is made against parasitic worms. Now, we keep talking about these worms. How does this worm stuff work? Well, I think I have a picture of it, and we'll talk about that too. But notice that the FC portion of the IgE is made against parasitic worms. Now that goes against everything that we've learned about. That's crazy. That's backwards. You have to remember the backwards stuff, okay? Because there are always exceptions, and this is one of the exceptions that, you know, people who live their lives figuring this stuff out say that this up here is this giant worm that's infecting a host. The IgE's FC portion, now it's not going to do a handstand on it, it's going to step on it. Okay? The IgE steps on the helmets, and we're all lined up stepping on this helmet with our hands up like this. Like, that's very strange. Arthropods, parasitic worms and arthropods that can bind to eosinophils, enabling opsonization. Okay, I'm going to show you the picture of it so in a minute, and we'll see how it works. Major defense against parasitic worms. Okay. Don't let me, don't. I know I have a picture. If not, I'll just have to act it out. Okay. These first two we've talked about. Opsonization. We know how antigens serve as opsonins. But let's act this one out too. All right. We said that macrophages have receptors for what portion of the antibody molecule? Phagocytes. Which part of the antibody sticks to phagocytes? The FC portion? Okay. Let's say that it's the FC portion. And let's see why that makes sense now. We have an immune response, and we have now made IgM and IgG antibody against this invader. Okay? Which part of the antibody is going to bind to the invader? The hands. Okay? This is our invader, so I'm going to stand on it or do a handstand on it? Handstand. We're going to do handstands. So all of us antibodies against this invader are doing handstands. Now we're calling in cytokines, calling in phagocytes to the area. Which part of the antibody molecule does the phagocyte have receptors for? You just said it. Come on, say it again.
FC.
The FC, my feet, right? Where are my feet right now? Sticking up. They're sticking up like this, so the phagocytes are going to come say, yay, look, there are all these feet. We can stick to the feet, right? The antibody's got the microbe, the feet are sticking up. I have receptors for the feet. It's now opsonized because I'm going to stick onto the feet, right? I'm a big phagocyte. I'm stuck to the feet. Now I can just wrap my whole little phagocytic body around it and eat it, right? Make sense? Anatomically, it does make sense. It sounds like, what in the world, this is so crazy. Why do we? But it's the way the immune system works. It's designed to work perfectly like this. Okay? So that's how opsonization of the microorganism by antibody enhances the ability of the phagocyte to come, get it, and get rid of it. Yeah?
Can you go back to the previous slide? Just what I looked up.
Okay.
I don't think that the FC portion of it.
Made against parasitic worms, okay.
It sounds like you said that the FC portion is the part that binds to the worm.
It says the FC portion of the IgE E, made against the parasitic worms combined, okay, yeah, go ahead. Okay, we're gonna show you a picture of it, and we're gonna, where if I have, confused that one, we're gonna unconfuse it. Keep me honest, okay? All right. All right, so, we see how that works. Agglutination, we said which isotype of antibody is really good at that?
IgM.
IgM, because it's got all these areas, got 10 places to do it. It can just grab stuff together and hold it tight until the phagocytes come. Again, the agglutinizing antibody is going to be stuck to the invader by how? During this agglutination thing, what part of the antibody is stuck to the invader during agglutination?
FAB.
The FAB. So what's sticking up?
Sepsi.
The FC. The feeder's sticking up. The phagocytes that are coming to clean them up, clear them out, is available. So it makes it easy for the phagocytes to come get it too. Okay. Neutralization. We haven't talked about this word yet. We talk about it mostly when our host, our patient, ourselves have been exposed to a virus or some sort of a toxin. Okay. Now, if it's a virus and our body has responded appropriately and it's made an antibody specific against the virus, the FABs, the fingers, grab the virus, and if the antibody has the virus can't do what it wants to do. The virus wants to find a perfect receptor on a host cell. It wants to bind to the receptor. It wants to get inside and take over the metabolic machinery of that cell. It's neutralized if before that happens, a specific antibody gets it. That's what's called a neutralization. Okay. Same thing if it's a toxin. We've been infected by a microbe. The microbe isn't what makes us sick. The microbe isn't what kills us. It's the toxin that the microbe makes. So we make antibody against the toxin. Once we have once we have antibody created in our bodies against the toxin, the antibody binds the toxin. So the toxin can't get into our neurological system. It can't do all the terrible things it wants to do to us. That's also called neutralization. Okay? So when we're talking about neutralization, we're mostly talking about viruses and toxins. I guess that last little bullet point sort of speaks to the toxin piece. Antitoxins, which is an antibody against the toxin that neutralizes the bacterial toxin so it can't do what it wants to do to make us sick. Okay? This we've already talked about how specific antibody does handstands on the invader. So they call it tag. It tags the thing that the macrophages need to come and get rid of. And that tagging promotes this opsonization, makes it sticky, and the macrophages themselves don't just have to get, kind of push them over in a corner and get them. They're actually bound to the organism that has antibody on the surface. Makes it really easy to get them in and do what they need to do. Neutralization of a virus. We can sort of think of this one as a toxin that may be in our circulation, and it can neutralize the toxin in the same kind of way. Agglutination, we said that the IgM is really good at this, and all these FCs are now available, and the macrophage can come over and make an easy lunch of agglutinated microbes as well. And it keeps the microbes from doing what it wants to do as well, which is multiply, get into deeper tissues, and do all those things and get destroyed. the very cool thing about some classes of antibody is that it can fix complement, and those classes of antibody that can activate the classical complement pathway are
IgG.
and IgM. And it can activate the MAC complex by what? The MAC complex starts with 5 what? A or B? 6, 7, 8, 9. Pick one. 6, 7, 8, 9. Yeah. And so makes holes in it, so destroys the invading cells. So these are all some of the things that our antibodies that we've learned about can do. Okay, there we go. This is yours. I'm going to clarify. Here it is right here. And the big element, it has the specific antibody bound to the surface, and the granulocytes come by and bind and don't eat them. Actually, remember we said that eosinophils believe that they are great phagocytic cells, and they do have some phagocytic activity, but they believe that they are able to eat these huge parasites. But what they really do well is not eat big worms. What they do is they bind and then they dump out lytic enzymes to put holes in the big cells. So the class of antibody that we're talking about here is what?
IgE.
Huh?
IgE.
IgE. Okay, so this is IgE that's bound there. The eosinophil comes and binds to it by its Fc receptor. It starts dumping out lytic enzymes to destroy the parasite. So in that situation, we're probably going to have more than 0.02 or whatever eosinophils in our circulation. But for the most part, they're almost undetectable.
Isn't it the same now? Because it does the handstand on the parasite, and then the
FC.
phagocyte comes to the FC.
Right.
Which is what we, so it's
Really the
not
same.
an exception.
Right, it's not an exception. They just make it appear so on the wording, but what it actually does is the same as it does for these guys. This is the only difference, is that it doesn't wrap itself around it. It sort of puts little holes in it by degranulating in the target area. Everything else we talked about. Okay, I think we've talked about this too. We talked about complement and which portions. We said for the most part it's C3b, but C3-4 is involved with opsonization as well. M and G are the best opsonizing isotypes of antibody. FC portions can bind to neutrophils, making the antigen, making it easier to stick onto the specific antigen. Another way IgG and IgM can be opsonizers themselves, and they can activate the classical complement to make the C3, B, and 4 elements that, so there's double opsonizing capacity of IgG and IgM. They can opsonize directly, and they can enhance opsonization by activating the classical complement cascade. Like IgG and IgM, C3b is a good opsonant, but you see, we keep adding C4b in there because it does have some opsonizing activity, but just not as good an opsonizer as C3b. And it just gives a few more words about how it acts. Once we get C3b to lock on to microbial surfaces, there are receptors on the phagocytes for the C3b that will allow it to come, lock on, and engulf and get rid of the invader. Okay, we talked about the glycoproteins, the slime layers, and the capsules being resistant to destruction by complement. It's really hard for the MAC complex to work because of the slime layers and the capsules. But because we do have the other elements, we have the C3b and the C4b that can attach to the capsule. Even though we can't make holes in it and explode them, we can still have some impact by complement against encapsulated organisms by making them easier for the phagocytes to attach to and destroy. We talked about this, we went through IgE against the parasitic worms, the FAB hands attached to the helminth, and the FC portion is available sticking up, not an exception, to the eosinophils and allows them, it says opsonization, but we know that those little eosinophils can't eat a big worm, but what they can do is that they can dump out lytic enzymes to try to put holes in it, and then they can eat the little pieces of parasitic worm that results. So they are phagocytic, but just not very efficient phagocytosers. Okay, what else? Promotes inflammation and recruitment of phagocytes. We know that because the IgE attaches to the granulocytes and can release the chemicals, the cytokines that induce